General properties of viruses. The viruses are smallest obligate intracellular infective agent containing only one type of the nucleic acid, DNA or RNA as their genome. They have no metabolic activity outside the living cells. They do not possess a cellular organization, lacks enzyme necessary for protein and nucleic acid synthesis. The viral genome nucleic acid diverts the host metabolism to synthesize a number of viruses specific macromolecules required for the protection of a virus progeny. They multiply by a complex process and not by binary fission. They do not grow in inanimate media. They are resistant to antibiotics. The major difference between the prokaryote and the viruses are shown in the table. Morphology of viruses. The viruses are small or much small than other organisms. The extracellular infectious virus particles is called as a viron. The size of the viruses ranges from 20 to 300 nanometer in diameter. The largest virus is the smallpox virus 300 nanometer, and the smallest is the pox virus 20 nanometer. The structure and symmetry. Structure: the viron consists of a nucleic acid core, genome surrounded by the protein coat, the capsid. The capsid together with the enclosed nucleic acid is known as a nucleic capsid. The capsid is composed of a large number of protein subunit polypeptide which are known as a capsomers. The certain viruses also contains the envelope that surrounds the nucleic acid. The envelope is lipoprotein in nature. The envelope viruses are susceptible to the action of lipid solvent like ether and chloroform. Symmetry: three types of symmetry are determined by the arrangement of capsid around the nucleic acid core. Isosacchariidal or cubical symmetry. An isosaccharidal is a polygon with 12 vertices or corner and 20 facets in the shape of equilateral triangle faces. This type of symmetry is found in Papua picorno adenoviruses, all naked or non-enveloped, and helps toga viruses envelope. Helical symmetry: the nucleic acid and the capsomers are wound together to form a helical or symmetry tube, spiral tube. Most of the helical viruses are envelope, and all or RNA viruses complex symmetry. Some viruses do not show either isosaccharidal or helical symmetry due to the complexity of the structure. These viruses are referred to have complex symmetry. Example: pox viruses. Shape: the overall shape of the virus particles varies in different groups. The pox viruses is brick shape, rabies viruses is bullet shaped, and tobacco mosaic viruses rod shape. Cultivation of viruses: the three modes are employed for the cultivation of viruses: animal inoculation, embryonated egg inoculation, and tissue culture. Animal inoculation: infant sucking mice are used in the isolation of arbo and coxsackie viruses, many of which do not grow in any other systems. Animals which may be inoculated by several routes: intracerebral, subcutaneous, intraperitoneal, or intranasal. After inoculation, animals are observed for signs of disease or death. Later on, they are sacrificed and the tissue are tested for the presence of viruses. The viruses are identified by neutralization test using antiviral sera. Some viruses inclusion bodies may be observed in stained smear beside mice. Other animals such as guinea pig, rabbits, ferrets are also used in some situations. Embryonated egg inoculation: embryonated hen's egg 7 to 12 days old or inoculated by one of several routes such as chorioallantoic membrane, allantoic cavity, amniotic sac, and yolk sac. After inoculation, the eggs are incubated for 2 to 9 days. Chorioallantoic membrane. CAM is inoculated mainly by growing pox viruses. It produces visible lesions. Pox. Each pox is derived from a single viron. The pox produced by different viruses have different morphology. Allantoic cavity. The allantoic inoculation is employed for growing influenza viruses for vaccine protections. Amniotic sac. The inoculation into the amniotic sac is mainly used for the primary isolation of the influenza viruses. Yolk sac inoculation. It is a inoculation for the cultivation of some viruses and certain bacteria, chlamydia, and rickettsia. Tissue culture. The three types of tissue cultures are available: organ culture, explant culture, and cell culture. Organ culture. Small bites of organisms are maintained in tissue culture for tissue culture growth medium. Organ cultures are useful for isolation of highly specialized parasites of certain organs. Example: tracheal ring culture for isolation of coronavirus air respiratory pathogen. Explant cultures, the fragments of minced tissue can be grown as a explant, which are originally known as a tissue culture. This method is rarely done nowadays. Cell culture. This type of culture routinely employed for the diagnostic virology. The cell culture are classified into three different types: primary cell culture, diploid cell culture, and continuous cell culture. Primary cell culture. These are normal cells freshly taken from the organs of animals or human beings and culture. They are capable of a very limited growth of culture, perhaps 5 to 10 divisions at most. Diploid cell culture. These are cells of single type that contains the same number of chromosomes as the parent cells and are diploid. They can be subculture from the limited numbers. After about 50 serial subcultures, they undergo sense and the cell strain is lost. They are also employed for the production of viral vaccines. Example: rabies vaccine produced in WI38 human immunogenic lung cell strains. Continuous cell line. These are cells of a single type that are capable of indefinite growth in vitro. They are usually derived from the cancerous tissue. These cells grow faster and their chromosomes are applied. They can be serially cultivated indefinitely. Therefore, they are termed continuous cell lines. Some cell lines are also being used for vaccine manufacturing. For example, vero cell line for rabies vaccines. Detection of virus growth in cell culture. The virus growth in cell culture can be detected by the following methods: cytopathic effect, heme absorption, interference, transformation, immunofluorescence, electron microscope. The nomenclature of viruses. The viruses are broadly classified into DNA, RNA viruses, and then further divided into families, subfamilies, genera, and species. Viruses can also be differentiated according to the genome and the presence or absence of envelope. Prions. Prions are infectious protein without any detectable nucleic acid. They are highly resistant to physical and chemical agent. They are resistant to heat 90 degrees centigrade for 3 minutes, UV rays and nucleases, and sensitive to proteases. They produce slow infections with a long incubation period, 1 in years. The disease caused by them includes scrapie of sheep, goats, mink, encephalopathy, kuru, and cruise-fed Jacob disease. For more details, refer slow virus diseases in chapter 2064.